Pharmacology Lecture Script. Title: Exploring the Complexities of Estrogen Receptor Modulators. Introduction. Welcome, medical students, to today's pharmacology lecture. In this session, we will delve into the intricate world of estrogen receptor modulators, ERMs. ERMs are synthetic compounds that interact with estrogen receptors, eliciting a wide range of effects in different tissues and cell types. Our goal today is to understand the various aspects of ERMs, including stereoselectivity, differential effects on ER-alpha and ER-beta activation, non-genomic signaling pathways, epigenetic modifications, and much more. Let's embark on this intriguing journey into the world of ERMs. ERMs exhibit stereoselectivity, meaning that the spatial arrangement of atoms in a molecule can determine its biological activity. For instance, some ERMs bind predominantly to ER-alpha, whereas others have a greater affinity for ER-beta. This selectivity can be attributed to differences in the ligand binding domain structure of ER-alpha and ER-beta. Understanding the structural basis for stereoselectivity is crucial for developing targeted therapies and reducing off-target effects. ER-alpha and ER-beta are two isoforms of the estrogen receptor with distinct tissue distributions and functions. While ER-alpha primarily mediates the classical genomic effects of estrogen, ER-beta is involved in regulating different cellular processes. For example, ER-beta activation has been shown to exert protective effects in the cardiovascular system and modulate neuronal survival. By unraveling the specific effects of ER-alpha and ER-beta activation, researchers can design selective ERMs with tissue-specific actions. Aside from their classical genomic effects, ERMs can also trigger rapid, non-genomic signaling pathways. These pathways involve activation of various intracellular signaling molecules, such as protein kinases and second messengers. Non-genomic signaling is highly relevant in tissues where rapid estrogen responses are required, such as the vasculature and the central nervous system. Further exploration of these pathways opens doors to novel therapeutic strategies targeting specific non-genomic actions. ERMs can exert epigenetic effects, meaning they influence the expression of genes without altering the DNA sequence. For instance, ERMs can recruit co-regulatory proteins to modify the chromatin structure, leading to changes in gene transcription. These epigenetic modifications can have long-lasting effects on cellular processes, including proliferation, differentiation, and cell survival. Unraveling the intricate mechanisms behind ERM-induced epigenetic modifications is crucial for understanding their therapeutic potential. Recent studies have unveiled intriguing interactions between ERMs and the gut microbiome. Emerging evidence suggests that the composition of the gut microbiota can influence the pharmacokinetics and pharmacodynamics of ERMs. Conversely, ERMs themselves can impact the gut microbiota, potentially altering drug metabolism and efficacy. Exploring this bidirectional relationship will shed light on the potential therapeutic implications of targeting the gut microbiota in ERM-related treatments. Developing ERMs with tissue-specific or cell-type-specific actions is a challenging but promising endeavor. Techniques such as receptor binding assays and medicinal chemistry approaches can be employed to design compounds that selectively interact with ERs in desired tissues. Targeted delivery systems, such as nanoparticle-based drug delivery, can further enhance the efficacy and minimize side effects of ERMs. A deeper understanding of tissue-specific ER signaling will pave the way for personalized medicine approaches. Structure-activity relationship studies aim to decipher the relationship between the chemical structure of a molecule and its pharmacological activity. These studies have contributed significantly to our understanding of ERMs by identifying key pharmacophores and providing insights into their mode of action. By manipulating structural elements, researchers can optimize the pharmacokinetic and pharmacodynamic properties of ERMs, enabling the development of more efficacious and safer drugs. Estrogen, and by extension ERMs, have been implicated in neuroprotective processes. They have demonstrated the potential to mitigate neurodegenerative diseases, promote neuronal survival, and enhance cognitive function. Understanding the underlying mechanisms of these neuroprotective effects will pave the way for developing ERMs as therapeutic options in neurodegenerative disorders and brain injuries. During the peri/postmenopausal transition, the decline in estrogen levels can lead to various symptoms and conditions. ERMs have emerged as alternative therapeutic options to hormone replacement therapy, HRT, for managing menopausal symptoms like hot flashes, vaginal dryness, and osteoporosis. Exploring the efficacy and safety profiles of ERMs in this context is crucial for providing women with appropriate treatment choices. Estrogen receptors are expressed in several immune cell types, suggesting a potential role for ERMs in modulating immune responses. Preliminary studies have indicated that ERMs can influence immune cell function and impact autoimmunity, inflammatory diseases, and cancer immunotherapy. Deciphering the complex interplay between ERMs and the immune system will allow for the development of innovative immunomodulatory therapies. Breast cancer is a heterogeneous disease, and treatment resistance remains a major challenge. Investigational ERMs, through their unique mechanisms of action, offer a promising approach to overcome treatment resistance in breast cancer. Novel ERMs that target different signaling pathways and engage in synergistic interactions with existing therapies may hold the key to improving patient outcomes. Estrogen receptors play a crucial role in regulating metabolic homeostasis, and dysregulation of estrogen signaling is associated with metabolic disorders such as obesity, insulin resistance, and non-alcoholic fatty liver disease. ERMs with tissue-specific actions hold potential in managing these disorders by modulating lipid metabolism, glucose homeostasis, and adipose tissue function. Investigating the therapeutic utility of ERMs in metabolic disorders opens doors to novel treatment strategies. In conclusion, estrogen receptor modulators, ERMs, offer immense potential in revolutionizing medicine. Understanding their intricacies, such as stereoselectivity and non-genomic signaling, and exploring areas like epigenetic modifications and tissue targeting will advance our knowledge of estrogen signaling. Embracing this complexity brings us closer to personalized and effective treatments. Thank you for joining me, and let's continue this fascinating journey together.